So what's new in Lasergene 16? We'll have a new navigator that we'll look at that's workflow centric. That means it'll guide people to the appropriate place within our software. We have the import of vector NTI data into SeqBuilder Pro. It's actually an improvement. We've had some of that functionality for several years. This is some new functionality that fits in better with the export function from their utility. And then we have some brand new functionality that we're quite excited about. Some PCR site-directed mutagenesis. It's really an enhanced way to do this. We can do some modeling and predictions on mutations that we enter. So we have some new algorithms for predicting how a mutant can impact the structure of a protein. And of course, that will involve both our SeqBuilder Pro and our protein tools. And we've added some modern NCBI BLAST and Entree searching capability that is very nice, a little wizard-to-do setup, and we can look at some results. And then we have some protein structure prediction. We've made some improvements there as well in our NOVA applications, and we're making these more available to people. So people who are trying out demos will have free access to running different predictions. So we're trying to encourage people to try and run some predictions, even if it's just for the fun of it. So the different Lasergene configurations, we've changed the packaging a bit. So for current customers, you'll notice that a little bit of the terminology has changed. We now have Lasergene molecular biology. And so this is our really kind of a core suite of tools for doing things like Sanger assembly and alignments and plasmid annotation and virtual cloning and primer design. We also have Lasergene protein, which allows you to look at protein sequences and structures and do predictive modeling. We have Lasergene genomics as well. So Lasergene genomics include things for assembly and visualizing NGS data. And then of course SeqBuilder Pro, which is really our most core application for molecular biology, and that allows us to do batch editing, sequence editing, primer design, feature annotation. So we have these 4 packages. We'll be looking at mostly the molecular biology and the protein tools today. And we'll go into the software then. And of course, as you get questions, feel free to chat them in, and we'll have another poll here coming pretty soon before we go into the protein tools. So, I can try to cover topics that you're most interested in. So at this point, we'll jump out of the PowerPoint. And launch version 16. You can see of course, this is my computer with all different versions on it. And when you first install, so for those new users, you'll notice that when you run the installation, a navigator will pop up. And the navigator's intent now is that, oh yeah, okay, good. So the navigator's intent really is to guide you into the appropriate places in our software suite. And we've changed it, so for those current customers, the navigator was really more of an enhanced start menu. It had a list of our applications, and you would organize by similar categories, and it had the product names for individual applications. And really, as we're moving forward, we're actually reducing the number of our applications and really focusing more on specific workflows. So, in particular, new users can find what they're interested in and be placed in the software at that point that they're interested. So for example, we have molecular biology, genomics, and transcriptomics, protein analysis and modeling, and then just a couple of utilities here, licensing the software and purchasing software, and then also suggest a workflow. And suggest a workflow brings you to our website. We tried to place all the workflows that we know our customers are interested in, but of course that's impossible to capture all of those. So we'd really like folks to provide some feedback, and you'll see that this will be somewhat fluid. We'll change some of the workflows, add some, maybe take some away that aren't used as often. But the way that this navigator works for molecular biology, if I select agarose gel simulation, it's going to launch SeqBuilder Pro and a gel. And so it gives me a nice starting point here for doing agarose gel simulation. And if you have questions on how to do anything, there's an information that brings us to a nice help. And the help is a combination now of tutorials and videos and screenshots. And so if you want to learn how to, do agarose gel simulation, that help brings you right to that particular workflow. So it's a very nice way to learn how to use the software. And depending on what it is that you're doing, if I'm in genomics, of course, we have a wizard. And so if I want to do a resequencing, SNP analysis workflow, it brings me right into that point for DNA-seq and genotyping. And this is, of course, our SeqMan engine wizard that will guide us through the setup of NGS assemblies. So a really nice way to get started. And we do use some different icons here that tell you which programs are going to launch. And so yeah, so it's a great way to start and be introduced to the software. So some of the new features. So that's the new navigator, and that will continue. Again, it's the first version of this workflow navigator. We're really looking for feedback from our users to try to fine tune it and add and remove workflows and put the type of things that you're looking for on that navigator. So we have some other functionalities too. The vector NTI import, which was recently vector NTI, the parent company Invitrogen is discontinuing support for vector NTI. And so what they did is they realized they still have quite a few users, and they have a proprietary database that makes access to that software, to the files, difficult, especially if you're trying to move to a different platform. And so they provide a utility on their website that exports the data. And this folder that I'm highlighting here, the VNTI advanced data, this is what it looks like. So when you run the utility, you can see that the DNA and RNA molecules are exported to a folder, enzymes, gel markers, the oligos, protein molecules, and some additional text files. So this is the file. So to import this, I'm going to go into Seq Builder, and there's a new file, import vector NTI data into project. So in the past, there were different ways to do this, and I can mention those as well. But so I can just select that folder, and it'll start to import the sequences. And what it does is it creates a multi-sequence file in Seq Builder Pro called a Seq Builder project file. And the project file then will recognize the folders, create those folders and the sequences that are in those folders, and maintain the folder hierarchies for you. And it takes us a little time just to bring everything in. And so now we're bringing sequences in, and it's asking me now, so the oligos are a little bit different. They're in the vector NTI database. They're in a comma separated file. And so it's asking me now, where do I want to save that CSV file? And so there, I'm just going to save that out. And so the primers then are in this CSV file. The DNA and RNA sequences are here. I'm just going to show you that I had gone in and actually created some subfolders. So you can see we had the subfolders for DNA and RNA molecules, some sequences that weren't in a subfolder. Here's the protein molecules, like that. And then if I want to look at the primers, here's the .pri. I'm just going to peek into it so you can see that there are some primers here added to this oligos folder. And you can see it's a primer, and the primer name, it's hard to read it like that, but we can bring this into Seq Builder. I won't go through all the steps, though, but I can bring in those primers into Seq Builder. So,
Matt, can I interrupt for just a moment? That was an excellent explanation of how to do the import. Once you're in Seq Builder Pro, some of our listeners might be wondering how to export that vector NTI data to the folder that you imported from. And we now have information and instructions on how to do that in the Seq Builder Pro help. If you go to the Seq Builder Pro help and just do a search, you don't need to do this right now, but if our users would like to search NTI in the search field, they should find very easily a topic linking them to the instructions on how to download their data from Vector NTI so that they will then have that folder they can import.
Great, thank you, Sharon. So that's the import, and as Sharon mentioned, there's additional in help under that topic if you have some additional questions there, and certainly we can help you. Now there was another question that came up about some archive files. So there is another method to bring in archive files, and that was before the NTI created this utility, and we do support that as well. And I'll just kind of show you the way that you would do that. In Seek Builder, I can open a new project and I can import. I just right clicked on that folder, and let's just look down here in the all known formats, and let's see if we have it listed here. Import. Oh, we have the Genius and Emble files, and I don't see it listed there. It's hidden, somewhat hidden. The old method will still work, is what I should say. So you can have, they're called our OA files, oligo archive, or DNA archive files. And that, if you have those kind of files, you can drag and drop them into this new folder. So we didn't remove that kind of older way to do things. So if you do have archive files, you will be able to just drag them into this project file and then export them back out. So we're going to move on from Vector NTI to some of the new tools in PCR site-directed mutagenesis. So site-directed mutagenesis is one of those workflows that our current customers have been using Primer Select in the past, and now it's Seek Builder Pro to design primers and introduce mutations. And it's really a great program for doing that sort of work. And of course, for many years, people have used these programs because it's got such a nice interface. And I'm just gonna, for doing that kind of work, and let me just open up a file here. So actually, before I do that, let's go back to it just a little bit to give you just a little bit of background on the data that I'm using here. And so this is our new protein design capability that's in Protean 3D. And what it allows us to do is a couple things. It can scan through regions of amino acids. And so it'll take a structure file, scan and look for what are called hotspots. It does a substitution of alanine at different, across those amino acids. And that scanning then can figure out by, which residues are the most impactful on the stability of that region of the protein in that structure. And so you can scan through proteins, find what look like important points, and then go in and create specific variations, in that protein and see what the impact is on the structure. And this uses a dead end elimination algorithm. I'm not a structural biologist, so I can't explain it much more than that. We have folks here that can certainly tell you in great detail what that means, but it's a pretty advanced algorithm where it does a kind of a continuous repacking of side chains, and it has backbone flexibility. So it's a very finely tuned algorithm then for predicting those structural changes. And what it looks for is a global minimum energy for that structure. So it's a confirmation then of the minimum energy for that structure. And there are some benchmarks there. So we used a couple different things. A case study where it was an E. coli lambda repressor protein, where it's the LLI variant, where it's in the core of that protein, some mutations were introduced to make it more stable. And so that's what we used for a poster that was at the recent PEGS conference. And we'll look at that same kind of sequence today, just for the demo. But there's also this FoldX benchmarking data set, and the FoldX data set is what we can use then to calibrate and make sure that when we make a prediction on a structure that it's in fact accurate. And we get a pretty strong correlation, a Pearson correlation of 0.7. So we feel that we've got between the wild type and the mutated form of the structure, a nice strong correlation then. And we included all the data. We didn't there are some outliers that we then we included everything, so even the ones that didn't behave themselves. So we have a nice correlation then on the accuracy then of these predictions. And so we'll look at this this E. coli repressor protein, and it'll kind of show you, we won't go through every step, but I'll show you kind of the feel for how we would do this. So we'll go into Seq Builder, and I'm going to open up the DNA sequence of this protein to start with. Let's see here, we have some fragments, and I have the and what I did is I annotated this sequence. So here's a piece of that lambda, and you can, maybe a little hard to see that, but you can see this feature here is the gene. It's this lambda DAP50 is the name of the locus tag for this. And what I did is I said, well, I want to introduce mutations. So I made these pink annotations at those places. So those are in the paper, those are the 3 mutations. And actually there's the protein I downloaded and the paper were off by 1 residue. So I might not have grabbed the exact right one. So everything's gonna be shifted by 1, but it's close enough for the webinar. So I annotated so I can kind of see what I've done. So I had designed a pair of primers then. We'll go to a linear view here and we can see, you know, here is this operon, and I have, you know, the repressor, the CL gene, and then some of the downstream genes from this lambda operon. And I'm going to design primers then right at those specific locations. So I added some annotations then that you can see. I can zoom in. So Seq Builder's got these really nice views where I can, you know, zoom in and see there's one of the variations there, and I can make them visible. I was doing a little bit of work on this one earlier, so, and let me get them right where I want them here. And one more. There we go. So there's different ways I can look at this. So here's kind of a linear map and the primer pair that I did earlier. Now how I made the primer, I'm going to go back to my sequence view or the primer design view. So primer design view is now what we used to call the primer workbench. And now I can see when I design primers, we'll have this view up. So we're going to design primers around, we'll zoom out here a little bit so you can see everything. So I can amplify the whole fragment. In this case is what I did earlier. And I just go to priming now and create primer pairs. And I'm going to choose primers exactly at this. So we need to design primers very specifically that are within, you know, a specific range. And so what it does now is it gives me this this primer, and I'm going to hide the one that I did earlier just so we're okay. So now I have a S2 pair 1 forward primer, and I can extend and retract the ends of my primer. So now I can modify the primer in Seq Builder, and I can choose where I want to introduce mutations. So if I annotate and I know where I want to introduce the mutation, I can introduce that mutation. And so I can click this ATC introduce mutation and go along and pick which amino acid I want to mutate to, and it'll automatically change that primer. And so that's how and we've had this functionality for some time, right? This has been many years where we've been able to go in now at each one of these codons, and I can introduce mutation. You can see the primer gets updated then. And that's how I can introduce. And now when I save this primer out, we'll have those modifications to the primer that introduced the mutation. Now what's new is that before I might have some other knowledge on where I want to introduce the mutation, but now we can do some predictions on the impact on that protein as well. And so I'm going to go to just minimize here. And I can go to my navigator again. You'll see there's a protein design and engineering workflow. And so I can make some predictions now on my mutations. And so we have, in this case, a benchmark protein where we know what some of the experimental results are, but that was done a few years ago and without predictive modeling. So we can even find additional models. So what's happening now is we have a protein design wizard that's coming up. And so I'm going to scan for hotspots first. And I can choose a file. And so this file then is a PDB file. And so depending on how you get that, you might, you can download it from PDB database. Protein 3D also has a file. If you know the PDB ID number, I can automatically open it. So there's several different ways to get started. New users come through the navigator. Experienced users may just go file and enter the PDB ID number. I've already done that, and I have the structure file now for that protein. And I can predict, there's two chains on the protein. I can predict and scan across both. And you can see that we're doing an alanine scan. We can also do a serine scan. And I can repack and allow backbone flexibility. This will make it take a little longer. I'm actually just going to turn it off just so it's not quite as, it'll take maybe 30 seconds to 2 minutes if I repack neighbors, but you can see there's some advanced options to really fine tune. If you want something that's just kind of quick and gives a result the fastest, we can turn those off. And we'll notice here while this is running, there's some new functionality as well that is in Protein 3D and Megaline Pro and GenVision Pro, is that we have this jobs view. And the jobs view keeps track of the things that I'm running locally here. So, and tells me things that I've done earlier, in this case, earlier today. So right now we're searching for the global minimum conformation, and I can see how close it is to being done. So it's running that scan. I can show my progress meter, which I think is this hiding right back here. I can also keep track of other types of jobs. So we'll look at some blasts and entree a little bit later in this webinar. And so now the job is done. And I can see that I've run a similar scanner. I'm just going to expand this a bit. So you can see I did the same thing this morning. And we save the jobs then. So as I run scans or do blast searches or do entree searches, we get this jobs list. So I can come in from another application, from Megaline Pro. I'll still see these jobs that I ran from the other DNASTAR application. So it's a nice way to start to pull things together and keep track of all the different things that I'm running. This interface is highly configurable. I won't go into that too much. What we have now is a structure and a report that comes back, which is the report 2, which is a hotspot scan for 179 different positions. And I'll just kind of scroll up and down, and you can see it scanned through all the amino acids and produced a defire energy potential and a prediction whether it's destabilizing, neutral, or stabilizing. So it's very, very fast. Went through all those amino acids just in a few seconds. We can filter this. I could say, well, I really am only interested in a prediction that's stabilizing. And now we've got a list of amino acids that are prime candidates now for introducing a mutation. And a structural biologist can dig into this further. I can select a region. So this first mutant, I can see where it is on the structure. I'm going to click this to kind of center it there a little bit and zoom in. And there I can see the side chain and where it's located on this protein. And these little filaments here are actually the DNA. So this is a DNA binding protein. So I can, in the Explorer, I can control what I'm looking at. So you'll see that as so there's a DNA filament that's highlighted in the structure view. So I can see, where the protein is in relation to where it's binding the DNA. So it's really a nice, and there's almost an infinite way I can adjust this view, to look at the different residues and look at the structure. The important thing is that I've got all these different positions now, so I can go in and decide both on just the energy potential. Maybe I pick one that's got the greatest D-fire energy potential. So I see, here's one at D14A, and I'll just jot that down as a, so I might decide that that's one that I want to target. And there's other ways to analyze the protein. So maybe I will do additional analyses and overlay those positions to try to find different regions based on secondary structure or antigenicity or hydropathy, to try to help me select which mutant do I think is the most impactful or going to have the desired result. So however you do your analysis, now I could choose to go back and introduce that specific mutation. And so I said D14A, and I'll just show you a different, I'm just going to close this now just to kind of show you a different way to, we don't need to save that. So rather than launching from the navigator, I'm going to open directly from PDB, and this is 1LMB. And so it's going up to the database and downloading. And okay, so there I have my structure again. Now, if I want to introduce a specific mutation, I can go back to modeling, protein design, and create a specific variant. And what I could do is select. So I can either introduce a document, or we can go into, see if I paid attention here to D. So there's my position there. So I just selected it in the sequence view. And now let's go back to modeling. Create a variant with the selection. And now I can choose, could choose alanine, or I could choose something different, a different residue, one that has, and however I pick that residue. And again, we'll try to make it a little, and so it ran a specific creative variant job. And again, I get a prediction on that particular variant. And here's where we could try and run, different amino acid substitutions. And this is, so I could go back and say, well, that prediction there, the D-fire energy potential, isn't quite what I want. Maybe I want to try that again. And so I think so I could go back and select and run that again, and keep experimenting. And here's where I'll experiment and just try different residues. And I don't actually know one that's any better than the next one, so and you can kind of see the process here. So I can run different variants, create those variants. And again, this is functionality that that for governmental and academics, this is free. It comes with Protein 3D, so it's not an additional. You'll see that this is available to you with version 16. And you can create variants and run predictions and hotspots.
Hey, Matt.
Yes.
We just got a question from one of our attendees, Tanya, and it's applicable to what you're talking about right now, so I thought I'd just go ahead and ask it now.
Sure.
Tanya would like to know if it's possible to run these calculations on a local PC or if you have to have a cloud server.
Very, very good question. So our protein modeling tools, some run in the cloud and some run local. the design tools are running locally. So they're very quick, and so most local computers will have no trouble at all, even running hotspot scans on larger groups of amino acids. So yeah, and we'll see right at the end of the webinar, I'll show you some of the NOVA that does run on the cloud, and you'll see a little bit some of the differences there. Okay, so again, we can keep working here, adding different amino acids, running a prediction till we find one that we want. And however we do that, we come back to Seq Builder. And Seq Builder gives us this interface. Now we have to go grab the DNA sequence, and we can now purposely introduce the mutation of interest, at that point of interest in our protein using this. And of course, if we're doing things like adding mutations to the interior of a protein, we may have a multi-fragment, and so I'll just kind of show you. I've got, in this case, an upstream fragment to the CL gene. So we may have an unmutated portion, so I added a mutation in the middle, and we have cloning then that will allow us to clone these fragments then into a single expression protein. So the way that works, I could just pick something like Gibson cloning, and then I pick a vector, and I don't really have one that's, well, let's see, which one did has at least an expression here. Here's one with GFP in it. And the way that these cloning wizards will work now is I need to set this up. So I'm going to bring in my inserts. And we want our mutated fragment, our upstream fragment I can bring in. And I select both there. And you can drag and drop. There's different ways to load these in, but I'm going to set the project. So I could have a lot of different sequences here, but to actually get them into the cloning wizard, I can drag them right into this surface here. So there's an upstream fragment. Here's a mutated fragment. And I picked a vector. I need to linearize it. I can see my green GFP protein there. I think on this clone, I think we want, well, I might not have. I think these are in frame, and this is where you'd want to have your vector and know exactly what cloning site. I'm making a little guess here. I think that is, so I'm going to linearize it there. And we're going to try it out. So we get this wizard then that shows us a little preview. We'll try it. And our cloning summary then is there's our vector, our 2 fragments, and it automatically designs. So this is PCR-based cloning. It will design the primers that we need, to do the cloning. And of course, we have instructional tutorials and videos that focus just on these steps. So if I'm going too fast, and here's our clone. And of course, the last step we'd want to do some verification. Oh, I put it in the wrong side of the clone. So I didn't mean to do that. So you can go back and pick a different site, and at the end validate, is it in frame or is it not in frame? And so I can go back to a sequence view in Seq Builder and do things like, show ORFs in, my top frames. And so, and by showing ORFs, I can figure out, let's see, top frames. Maybe the linear map is better. So I can try to figure out, did I clone this in frame or not? And I got my fragment up ahead of my GFP. I picked the wrong enzyme there. Oh, it was the EchoRV is what I wanted. That's why. And so we can get this in frame and validate that everything's in frame, and we're good to go. So again, it uses functionality, this primer design workbench that's been in Primer Select and in SeqMan for many years, but now we have this predictive capability. So the real enhancement is now in Protein 3D, where I can use modeling to guide where I want to introduce my mutations. Okay, so that is, now I think we do have we have a third poll that's coming up? I have not covered search. We can go through that next, certainly. All right, we've got a few minutes yet here, so I'm going to go to another piece of new functionality, and I'll open Megaline. And in Megaline, I have just a project here of some demo data, some calmodulin sequences that I've aligned with a muscle algorithm. And you'll notice now that we've got in Megaline Pro some search. And search is both sequence-based for sequences or for text, an entree type. I can select regions, for example, go to search for sequences. And this brings up a wizard now. So our older applications didn't really provide much guidance. You would select and just click search and it would run a job, and you would wait and it would just kind of appear. And what we did is we made it a little bit more like NCBI, where now I have some more control over my searches. And so I bring up a wizard and I can search it. It should recognize if you have proteins or nucleotides. And I can pick, in this case, it's going to be BLAST-P, and I can pick the database that I want to search against, right? And I could run the job now, or I can go into the wizard and change some of the parameters. And so sometimes with matches, I might want to limit the number of sequences that come back, and I can put some limits on the search results that are returned to me. So a little bit more flexibility, and I can name this job now. So we'll call it and I click run. And so it'll upload those files and I get this progress meter. And you can see then I can hide this and I can continue working, and I can see the job here is just starting up. So calmodulin search 2, if I hover over it, I see, you know, that it's just a running job, and I get the status. All right, and now I ran the same search earlier today. So the nice thing now is that they're not ephemeral, the searches. They will be maintained in this jobs view, and I can select jobs and delete them if I need to, or I can just hang on to them. So it's sometimes nice to go back to searches that you've run. And here's a result then, and you can see that it's quite a bit more colorful than it used to be. So we have a list of the jobs. There's a pull down at the top that if you have a multi-sequence query, you can select the sequence, the query sequence, and then go through, and you'll see that we have a pairwise alignment below. I can use my arrow key and quickly go through these results, and we get e-values and scores, and we get a little map that shows us how similar, so it's styled more similar to NCBI. I get a text output as well. So the bottom view, we have pairwise view, and then there's also a text view. Right, so this is more of the traditional kind of output that I could save out to a text file. And then of course, with this different output, I can do different things with it. I can filter. So and I won't go through all, this is kind of basic filtering to try to narrow down my results. I can also save, and I can actually save the search result. And so if you've run a search that took, sometimes a search can take quite some time. If it's a large search that is more elaborate, I can save that search out, or I can do stuff, with the sequences. So I click this icon, I can add the sequences to this project, for example. So I could select a sequence. Let's say here's corn, calmodulin. So now I'm grabbing that sequence and adding it right to the Megaline project. So that's, and we can see the download in progress here. And so as you download sequences, that also goes into the job query. So if you're downloading large numbers of sequences, or it's nice to kind of see that progress. And here's that sequence I downloaded, so I could add that into my alignment. So it's a very handy search interface with nice features on it. Now I can also do a text search, and I'll just, and of course this is more of just an entree type. It is an entree type searching, so I can, all fields contains, and again, we get that job list. And you'll see this, so this new search capability is in our newest applications, and you'll see it eventually added to Seek Builder Pro, and some of our other applications will continue to get feedback from people. So you'll see we have on all our new in our software, a little suggest features. So again, we encourage everybody, if there's some piece of functionality that you can't find, certainly let us know, and we'll do our best to add that. So the last little piece here, I'd just like to mention, we're going to go back to the navigator here and the protein analysis and modeling. And you can see one thing I maybe didn't show is that when you hover over the workflows, you get a little bit more elaborate description of what they are. So antibody modeling, protein design, if you're wondering what they do, you can just kind of hover, or again, go to the information that'll bring us to our help that has tutorials. And for many of the different topics, there's videos as well, right at the top of the intro videos for these different workflow topics. One last piece I want to show you is just some of the structure prediction. So all of these modeling tools, so prediction, docking, antibody modeling, they will run on the cloud. So if I do a structure prediction, and now I'm logged in as myself on this computer, so it will recognize that and bring me to this page. And so the things that run in the cloud, the Nova services, bring me to this kind of a wizard that allows me to set up what I'm trying to do. Now, if you're a new user or a demoee, you'll be asked for your login credentials. So it'll be typically your email and the same login that you use to access your demos. And so keep track of those emails, if you're just starting out, and we can, so you can access the cloud, and all our demoees will get some predictions with their demo. And so you can try out NovaFold and NovaFold antibody and NovaDoc. The protein design runs locally, so you'll have unlimited for introducing a variation. And so we'd encourage you, try these out, and you can use some of the proteins that are provided in our demo data, or you can predict your own. So for NovaFold, I'm just going to click on this NovaFold, and there's been some improvements made here as well. There's more. The algorithm now is more sensitive to clashing between molecules. So we've made some, and we're doing some new benchmarks on that as we speak. So you'll see in our marketing materials more benchmarking on the accuracy for our structure predictions. And so to run something in the cloud, again, you enter in your credentials that are for your customer account that are sent to you via email, and we can just add a file. So we'll just go back to a commodulin file. And you can see it's just a simple wizard here. And there's some I could just submit, or I could go in, an advanced user, could go in and change how many models they have or add constraints. So there's some advanced functionality for the structural biologist. So I just keep everything at default. There's my job name and submit. And so now this is going to be done up on the cloud. And we do have for the fold, we have and if you need to run this locally, you can contact us. We have some options now for running NovaFold locally on a Linux or a Mac computer. So that's something you can, if you're interested in, just contact us and we can help you with that. And you can see now all this is running. This is on the cloud, so it'll be tracked here, we have a predictions panel, and this will just keep track of jobs that are running up on the Nova services. And you can see I've run barley calmodulin earlier today, and one that we just started. And so we can go to these jobs here. And let me just minimize that. And so there's just a list of the different jobs that I ran. It looks like we have a NovaFold antibody job that had run. So this will build this list as you run these predictions. And again, we can go back to the models, open them up in Protean 3D for analysis. Okay, well, I think, let me go back. I think that's, yeah, I'll just open up this last slide. So again, if you're a new user, there's a trial that you can download. We have SeqBuilder Pro. We have some nice new pricing, low pricing, as low as $99 a year. And if you have any questions, there's my email. You can email me directly. And we'll have this webinar online in the next couple of days. And with that, I'll take any questions that have come through.
Okay, next question is from Ali, and he's interested in the protein design workflow. So he says, Matt showed us that by introducing a mutation, we can see the prediction of energy related to protein stability. However, I'm also interested in knowing if we can predict the impact of the mutation on the substrate binding site of the enzyme or on its regioselectivity.
Right. Yeah, great question. We don't have anything to predict the substrate binding other than depending on what the substrate is. If it's another protein, we have docking algorithms and antibody modeling. So we could introduce a mutation and then look at its ability to bind other proteins. So that's a possibility. If the substrate is a small molecule of some sort, we do not have that prediction.
Okay, he says the substrate is an amino acid. He just
yeah.
Okay.
Then the docking, yeah, we certainly can do both, introduce a mutation and see how it affects docking.
Okay.
Absolutely.
And let's see here. I think the last question is, if I'm not mistaken, that I was not able to answer on my own. Okay. When should somebody use the start menu to open the application versus using the navigator?
Yeah, a great question. So I'll go back to this navigator here. If it will let me escape this, there we go. And so the navigator really has been tailored for new users now, where you might not know which application to launch for specific functionality. Once you're familiar with the workflows that you have, then start menu is probably, the best place to go. If we get enough feedback from our users that they would just like to switch the navigator over to a list of applications, that's something that we could add to this navigator, add back at some point in the future.
Okay, and let's see here. Oh boy, the questions just keep pouring in. And I guess we'll just handle this one more.
Sure.
Michael asks, is there a small molecule docking function in Protean 3D or any of the Nova apps?
I'll say not yet. That is a frequent question that we've gotten at different meetings from customers. We do not have small molecule. It is certainly on our radar.